Thank you, Alain, very much for the invitation. And you're going to listen about dopamine and the control of neuronal circuitries. And since dopamine is rewarding and we are late, I don't want to do a big experiment of food restriction, and so I'll try to be as short as I can. So what we have heard until now are disorders that are associated with the dysfunctional dopamine. And in these slides, the only thing I wanted to point out is that dopamine in a different circuitry of the brain, whether it's either down or up regulated, the level of dopamine can induce, lead to neurological and neuropsychiatric disorders. So we have heard about Parkinson's disease, which is due to a reduction of more than 80% of the dopamine content of the brain. So from these, what appears clear that although huge steps that we have made in studying the dopamine system, there are still many questions that needs to be addressed, and by far we have no idea, like we have heard from Alessandro this morning and others, that we still don't know the etiology of most of these neurological and neuropsychiatric disorders. So my lab has been involved in understanding how dopamine works, and in particular focusing on dopamine receptors. In fact, whatever dopamine does is mediated by the interaction with the 7 transmembrane domain G-protein coupled receptor. During the years since their cloning, people has been generating mutants for each one of these receptors. And what appears clear that single mutants are, animals are able to survive, and D1 and D2 mutants have most important phenotype, which is likely due to the fact that they are widely expressed in all areas that are innervated by dopamine fibers. However, D1 and D2, as I said, they are very important, and in fact, studies in the lab showed that if we now make a double D1, D2 mutant, animals do not survive after the 2nd week of age. So clearly, whether you do not have dopamine or you do not have D1 or D2 receptors, this is not compatible with life. And in fact, similar data were obtained by the Palmiter's lab in animals that don't have dopamine. So as I said, the topic of my laboratory has been the study of the dopamine D2 receptor. Dopamine D2 receptor are very important and I think are critical elements of the dopaminergic system. One important thing is that D2 receptors are expressed presynaptically, and presynaptically they control the release and also the synthesis of dopamine through the inhibition of tyrosine hydroxylase activity, through the reduction of the phosphorylation site. So presynaptically, dopamine controls dopamine, and more recently it has been shown that these neurons can also release GABA, and GABA is packed in the same vesicle that have dopamine, so D2 has this important function of regulating release from these neurons. And also the activity, and in fact D2 stimulation, hyperpolarize these neurons. At the postsynaptic site, D2 is also present, so we have this dual expression of these receptors, and postsynaptically they inhibiting, for example, the firing of striatal neurons, but they also have heteroreceptor function, of which I will tell you in just a few moments. So if we now take the striatum and the dopaminergic neurons, so you have heard a great deal about the substantia nigra and VTA, and so D2 receptors are expressed here, but D2 receptor, if we take only the striatum, are also expressed in the striatum, in the caudate putamen, and in neurons that are the medium spiny neurons, which are the major and only output of these structures. In particular, if we now think about motor activity, which is stimulated by dopamine reaching the cortex but also the striatum, is regulated by dopamine interacting with medium spiny neurons expressing D1 receptors, and these neurons project back directly to the substantia nigra reticulata that they inhibit, but also through the indirect pathway, which is dominated by D2 receptor, and D2 receptor inhibits the firing of these neurons. So it is thought that a balance between a direct and indirect pathway is extremely critical in the control of the output to the thalamus. And in fact, the substantia nigra reticulata inhibits the thalamus, and then the thalamus projects to the cortex and striatum, and that's how motor activity gets regulated. This is, of course, a very simplified circuit of this function. So in addition to what we would just, I show you, which we can call like a macro circuit of regulation, there are also micro circuits inside the striatum. And in fact, D2 receptors are expressed, as I said, on medium spiny neurons and on dopamine neuron terminals, but they're also expressed on terminals from corticostriatal fibers as well as on cholinergic interneurons. And it is, we believe, the interaction between these macro and micro circuits that regulate most of the functions that are associated with dopamine signaling. The first thing that we did, and this was a long time ago when after having cloned this dopamine D2 receptor, was that we achieved the knockout of this receptor. And it's interesting because this knockout presents major phenotypes. So if you remove D2 from the brain, we got a Parkinsonian-like phenotype. So these mice are not able to move very well. They move like 50% of their control, wild-type mice. And also, they cannot coordinate movement. In addition, we showed that absence of D2 leads to a loss of regulation of dopamine release. And also, the motor and rewarding properties of drugs of abuse, which are mediated by the mesocortical and mesolimbic pathway, are affected, and these mice develop pituitary tumors. So as you can see, absence of D2 affects most of the functions that are associated with dopamine signaling. So to go back to the microcircuit, what we believe is the reason for that, and this is our hypothesis, is that D2, by regulating dopamine release, regulates the signaling in medium spiny neurons through, for example, D1 receptor, but also on cholinergic interneurons, modulating how much acetylcholine is released, and GABA, as well as glutamate from corticostriatal fibers. So in the absence of D2, what is happening is that all these neurotransmitters, and we don't know now to which respect, because we did not have at the time a very refined models, we believe that that's the reason why these D2 knockout mice recapitulate most of the dopamine-mediated function in our models. So in order to really dissect these functions that D2 has, we have taken the conditional knockout approach. And we have deleted D2 specifically in dopaminergic and medium spiny neurons. And I will show you some of the data that we have obtained, both at the level of motor activity and also from the mesolimbic dopaminergic system. So here you have an in situ hybridization with D2 showing the substantia nigra labeled and the striatum. And you can see that if we remove D2 from the dopamine neurons, we don't have any longer D2 expression here, but there is a persistent expression in the striatum. And vice versa, if we delete D2 from the medium spiny neurons, we preserve expression in the substantia nigra and VTA, but not in the striatum, with the exception of these little dots that represent the cholinergic interneurons. So the first thing that is interesting that these mice have told us is the contribution in the striatum, for example, for PET imaging and other techniques that are normally used. What is the contribution of D2 receptors coming from dopaminergic neurons? And so the analysis of these mutants that don't have D2 on dopamine cells shows that if we take a striatal extract, 20% less D2 receptors are now, so 20% of the receptor that we measure in the striatum, 20 to 30, are coming from dopamine fibers. On the other hand, if we delete D2 on medium spiny neurons, we get a reduction of 70% of binding sites. Now, so what happen when we remove D2 from medium spiny neurons? And these results, those are behavioral experiments where we monitored motor activity and coordination, and this is a rearing activity, so the coordination of movement. These two functions are greatly impaired when D2 is removed from medium spiny neurons. So this is a function that we now can feel confident to say that is mostly dependent on medium spiny neurons. And in fact, this phenotype resembles that of the full knockout that I just told you have a Parkinsonian-like phenotype. The reason for that, we believe, is due to the potentiation. The absence of D2R from the medium spiny neuron results into a potentiation of the indirect pathway and a stronger inhibition of the thalamus, and so a lower activation of the cortex and striatum. Now, conversely, when we remove D2 from dopamine neurons, we observe exactly the opposite phenotype. And these mice have a higher motor activity in an open field and more vertical movements than their control litter mates. This again matches with this model in which by removing D2 from the substantia nigra and VTA, this should lead in most of the region, here I have highlighted all of them, but we for sure know in the striatum that there is more dopamine that is released and stronger activation of the striatum that leads to a potentiation of the indirect pathway and then a stronger activation of the thalamus and the projection back to the striatum and cortex. So this is at the level of the macro circuits. If we now look in the micro circuits, as I told you, dopamine D2 receptors control the activity of tyrosine hydroxylase through the cyclic AMP pathway, and in fact, D2 receptors are inhibitors of this pathway. And when D2 is activated, the phosphorylation on serine 40 is greatly diminished. So what we did was to take punches from the striatum, so where the dopaminergic fibers arrive, in the dorsal striatum and nucleus accumbens of mice that do not express D2 in the dopamine neurons. And what you observe here was, with our biggest surprise, to notice that the fibers that projects to the nucleus accumbens clearly show the absence of the D2 autoreceptor. And in fact, tyrosine phosphorylation is increased and is no longer controlled by quinpirole in the nucleus accumbens. However, when you look in the dorsal striatum, this is not the case. And this was for us very surprising and showed also that the dopamine release in the dorsal striatum is not only controlled by the autoreceptor, but also from D2 receptors present on other neurons, striatal neurons, like, for instance, the cholinergic interneurons, because this interaction between choline, acetylcholine and dopamine, it's very now be described, it's very clear, and there is really a dual control from the dopaminergic neurons of these cells and back to the dopaminergic neurons. So clearly the absence, and while in medium spiny neurons knockout, we do not see any effect on tyrosine hydroxylase expression. So clearly these mice have helped us in identifying this type of differential effect of D2 on neurons from the substantia nigra or the VTA. Now, let's look at another pathway that is important, that is the mesocortical limbic pathway, in which dopamine controls reward. And reward, this food intake is a classic natural reward. And this pathway is hijacked by drugs of abuse, of which I will not talk about today. But so let's look at food intake. Food intake is something that is very, again, very interesting. In the full knockout, this is the total knockout, we observe that the body weight of these mice is a little bit reduced. And so it is that of mice that do not have D2 in medium spiny neurons. However, those that are missing D2 on dopamine neurons are perfectly normal, like wild type. Now, if we look at food intake, you can see that in the full knockout, we get 21% decrease of food intake, and in the MSN knockout, we get a 34% of reduction. So, while no reduction is observed when D2 is removed from dopamine neurons. So this can be justified by the fact that I just told you that these mice don't move as much as wild type. So they move less, they eat less. However, we made this food intake and body weight ratio, and it was clearly a strong reduction of the weight, like of these parameters in these mice. And so we asked, are these mice able to eat palatable food? Because as I said, dopamine is very important in the rewarding mechanism of the brain, and the palatable food generates, and that's why we eat chocolate, fat, and so many other things that we say we like it just because it's rewarding. So this is this is a short movie which I thought was fun to show. This is a mouse that, without any food restriction, this is a wild-type mouse, has been offered peanut butter. We are in the United States, so we put peanut butter here. And you can see that this mouse, this is a wild-type mouse, will eat this food. They really like it. And then it will eventually move away from the food. Now, this is an MSN D2R knockout mouse. And you can see that these mice get crazy for palatable food. So they eat way above the wild type. And these guys, you can hardly take away the mouse from the peanut butter. And so when you put this on a graphic, which is less fun, but if you put the preference, you can see that mice deleted for D2 on dopamine neurons do not differ from wild type. However, either the deletion from medium spiny neuron in the full knockout, either the full knockout of this or deletion of D2 in medium spiny neurons lead to this preference for food. And the preference for food is interesting because mice, unlike humans, they control their caloric intake. And so these mice go well above that. And in fact, with time, they get fat and they start to develop glucose intolerance. So this is an extremely interesting point because it shows that the presence of D2 in the striatum is controlling this important part of food intake, which is very likely linked to the fact that in addiction, for instance, and also in obesity now, it has been shown that there is a reduction of D2 in the striatum. And so these mice kind of simulate what has been observed in humans with a reduction of D2. And very likely shows that they cannot, so absence of D2 on medium spiny neurons kind of prevent them to control the intake. And right now, and I don't have time to show you all the data, but we have localized this function in the dorsal striatum, and we found connection from the dorsal striatum to the hypothalamus. So this is a new type of pathway that we have discovered and that we are right now characterizing. So in conclusion of this first part, what I am telling you is that ablation of D2R on dopaminergic neurons reveal a differential regulation of dopamine synthesis in the dorsal striatum and nucleus accumbens, and that the D2 autoreceptor inhibit dopamine neurons firing. And this is an important point for dopamine receptor aficionado, because in the substantia nigra there is also D3 receptors, but what we show is absence of D2 cannot be compensated by D3-mediated function. Conversely, ablation of D2R on medium spiny neurons reveal a critical role of D2 in regulation of motor function in medium spiny neurons, and extremely important in the regulation of food intake, and in particular also in limiting the amount of palatable food that is eaten. Now, in the last part of my presentation, I just want to summarize very interesting data that we have observed by analyzing these dopamine D2 knockout mice, and whether these mice can be a model for psychiatric disorders. Why that? So if we look just at the behavioral data, you see that these mice, like I told you before, they have an hyperactivity in the open field. This is the motor and this is the rearing. Also, these mice respond much more to amphetamine than wild-type mice. So you can see, for example, at this dose of 1 milligram per kilogram, these mice have a higher response in motor activity than their wild-type, but also at a higher dose. Now, the hyperactivity in a novel environment and hyperresponsiveness to amphetamine are two behavioral phenotypes that are classically used to model psychosis in rodents. So what we did, we took these mice and we performed, because, you know, like if they are a good model of psychiatric disorders, and in particular we are thinking to schizophrenia, or psychosis, these mice should have an affected working memory. And this is the case. You can see here, these mice were exposed to a radial maze where only 4 of the arm were baited with the food. And you can see that the time to perform is highly impaired in these dopamine D2 receptor KO mice. And also, this is the performance. The performance is less good than in wild-type mice. So clearly, they have a working memory that is affected. Then we went on further, and we assessed the sensory motor gating in these animals by performing the startle reflex and the prepulse inhibition. And you can see that in the startle reflex, these mice behave differently from wild type, again, you know, in line with the good model to model psychosis. And similarly, the PPI is also affected at this sound level. So what we thought we would do was to analyze gene expression in these animals in the mesolimbic pathway. And so we took mRNA from the ventral striatum and from the prefrontal cortex, and we performed a microarray. In the microarray, this is the result. This is a heat map of the nucleus accumbens, and you can see that there are genes that change. We found at least 100 genes that are differentially expressed in this area of these mice, in the nucleus accumbens. However, when we looked at the prefrontal cortex, that's where we found this amazing difference in gene expression. And we found an outstanding downregulation of over it's 1,900 genes in the prefrontal cortex of these animals. And so this massive downregulation of gene expression led us to think that this could be due to some kind of effects that are mediated by dopamine at distal site, so the dopamine that is no longer controlled in their release in the prefrontal cortex. And one mechanism could be an epigenetic mechanism. And so here an epigenetic mechanism due to the fact that, for instance, here you see that the geoanalysis of these genes showed that there was a majority of genes, oh no, sorry, this is the ventral striatum, this is in the prefrontal cortex, so you see that the majority of genes are belong to the regulation of transcription that is DNA dependent, and some of them to chromatin remodeling. And so what we did, we analyzed this prefrontal cortex by performing a mass spectrometry, but also by immunofluorescence analysis. And what we found, among many other modifications that we tested, was an upregulation of H3, of histone H3 lysine 9 trimethylation and demethylation in the prefrontal cortex of these animals. This was done by immunofluorescence, but then it was also confirmed by mass spectrometry in collaboration with Axel Imhoff in Munich. This is very interesting because at least we are very excited about this data because, as you know, schizophrenia is a very complex human disorders. And there are many genes that have been associated to this disorder, not a single gene, and which could be justified by the fact that it's the whole circuitry that is affected, and very likely dopamine can contribute to that. And in particular, even more important, is that the D2 receptor that we have been removing from dopamine neurons is a target of antipsychotics. So it's a pretty good story, and I think this is a very good model for the disorder. More importantly, this effect that we observed on histone H3 canine methylation appears during the adolescence, which is another feature that is common to human disorders like schizophrenia, where it appears during the adolescence and then it persists in the adult. So what we did was, more recently, was to treat these animals with the classical antipsychotic. So we used clozapine, and we looked at two things: the behavioral effect of the antipsychotic treatment and also the cellular effect. So at the behavioral level, of course, you are giving an antagonist of D2 receptors, so you observe a reduction of motor activity, and this is true in both genotype, in wild type, and the knockout mice, okay? And so clozapine is able to reduce the hyperactivity in an open field. However, at the level of the prefrontal cortex, clozapine is it cannot revert the H3 canine trimethyl, and also here, I think, yeah, this is a quantification of this same data. So clozapine is inefficient to do that. So then we did the D1 and D2 agonist treatment, and I just show you the D2 treatment, which is the one that worked really well. So now, if we take these animals and we treat them chronically with quinpirole, a D2 agonist, we are able to revert the phenotype, the motor phenotype, but also the PPI. So you can see here that these mice can be rescued by quinpirole treatment. And similarly, here you can see the effect by Western blot analysis. This is the quantification. And also on two genes that we had identified as to be downregulated in the prefrontal cortex of these animals, which are AKT and NUR1, also called NR4A2. So these are two genes that have been associated with psychiatric disorders. And you can see that the quinpirole treatment reverses the downregulation of these genes. And to finish, we are also looking into some, these are a completely biased choice that we made. These are histone demethylases that are also downregulated in these animals, and we can see that quinpirole can reverse the downregulation of these genes. However, now, as an approach, we are doing the chromatin immunoprecipitation to isolate what are the targets of this modification in vivo. But overall, what I think these data show you, and what is important, is that loss of D2 receptors in dopaminergic neurons affects the epigenetic landscape at distal sites. So this is an important, I think, finding, because simply a neurotransmitter, and probably dopamine is not the only one, but an alteration of dopamine or serotonin or any other type of, maybe not, but I mean for dopamine for sure, leads to major epigenetic modification. And this leads to an increase of repressive marks at the chromatin level and downregulation of gene expression and altered behavior. And this, I think, I would like to conclude by saying that hopefully in the future, we will be able to pinpoint what are the genes that are really targeted by this modification. And so to finish, I want to thank Karen Bramicherier and Andrea Anzalone that performed most of this study, while the food intake has been done by Daniela Rado, and then all my collaborators, and Wolfgang that is here for giving us the Cray line, one of the Cray line. Thank you very much.